Hi there. In this tutorial video, I'm going to show you how to use differential expression analysis through the scatter feature of DropViz to identify markers both at the level of global clusters and at the level of subclusters. And for this tutorial, we're going to focus on the frontal cortex dataset and neurons, class neurons within the frontal cortex dataset, which I've loaded here. And again, we're looking at the color-coded t-SNE plots of the global clusters for neurons in frontal cortex. And for the purposes of this analysis, I'm going to compare the two interneuron subclusters, cluster 1, which are the complexin synperin expression presumed CGE derived interneurons, versus interneuron cluster 2, which are the somatostatin parvalbumin expressing presumed MGE derived interneurons. Okay? So, to do this level at the global comparison of clusters, we want to make sure we're in the global clusters tab, and we're going to go into the clusters tab here, and you'll see this window called compare clusters. And what we're going to do is first select the interneuron cluster 1. Okay? And you'll see a bunch of genes now have popped up, and these are all the genes that we found to be significantly enriched in cluster 1 versus a synthetic version of the bulk tissue data set. And you can see that more clearly by clicking on the scatter tab. And what the scatter tab displays is a scatter plot of gene expression. Now the scale here is normal log, and we're looking at the average gene expression. And you can see our target cluster that we've defined here is on the x-axis. And for a comparison, as a default, we're using the rest of the frontal cortex here on the y-axis. And you can see a number of lines here, and I'll explain what those are in a minute, and a color-coded p-value, and that has to do with the p-value of the statistical test we use to derive significance here. And any genes that are above threshold for the defined p-value, and I'll show you how to change those parameters, show up as dark dots. And those dark dot genes are shown here in a table below. And this table is very convenient. For example, we can sort it. So if we're looking at it for the genes that are like most enriched, we can sort it by fold ratio or by p-value or the comparison values. Okay. So let's we'll sort by fold ratio here, and we can look at VIP. And so if we click on the row associated with that gene, we can see the gene is now labeled in the scatter plot. So you have a good sense of where these markers actually fall in the test. Okay. Now, you're limited here to the genes that are defined as statistically significant. And again, I'll show you how to change those parameters. But before I do that, I wanted to say that you can force a gene to be displayed on your scatter plot of interest by putting it into the gene tab. So for example, if I'm interested in actin B, I'm not sure if that's going to be differentially expressed, and indeed it's not. It seems like it's quite highly expressed in both cell types. And you can see now actin B is associated with a red dot because it doesn't fall within this thresholding of statistically significant genes. So how do we adjust this thresholding? So if we go to the clusters tab again, you can see this is where we identified our comparison cluster. And then you can see that we have differential expression criterion here. And so, for example, if we wanted to, and a number of different tests, right? We can ask for genes that are above a certain minimum fold ratio, that have a threshold maximum p-value, and have certain levels, required threshold levels, in our target cluster versus the rest of the data set. So for example, you see this line here is controlling how much of the significant genes we tolerate in the synthetic frontal cortex, the max mean log in the comparison. And so if we wanted to tolerate more, for example, 3, we can drag now it over to 3, and we can see this line here has gone up, and the number of genes that fall into this window has also gone up. Okay, so here are all the genes. And that's because we have the AND function here. So we're requiring a minimum fold ratio of 2, a maximum p-value of 100, and we want all the genes to kind of fall into this window here. Okay? So we can, I'm just going to lower this back down to 1. Okay? And this, for example, in the minimum mean in the target, is asking like what threshold we need in our target of interest to be considered positive. So if we wanted to drop this down to, say, 1.25, then we would again collect significant genes that are on this side of that thresholding. So this whole analysis really focuses on finding markers for a particular target cluster versus some other comparison. So for example, now if we wanted to make that comparison, instead of a synthesis of the bulk tissue, the cluster number 2, the somatostatin and parvalbumin interneurons, we could click on that, and again, now everything is recomputed on the fly, and we will find a new set of comparisons here. Many of the same genes are positive by this test. Okay, so so you should definitely play around with the thresholding here to identify significant genes. One other nice feature, we can just look at fold ratio only if we want to find markers that are associated with either of the two comparisons. Okay, so now instead of just focusing on interneuron cluster 1, we're also looking for markers for interneuron cluster 2. And again, we can adjust the fold ratio. So for example, somatostatin, as we'd expect, is high in cluster 2. The VIP is high in cluster 1. We can, again, adjust all these thresholdings to map the genes that we're interested in onto the scatter plot. So that's all well and good. And we can do a similar comparison, say, for example, within subclusters. So this is, maybe many folks will be interested in this type of thing, where now, by selecting the subclusters tab, we have all the subcluster options involved in those two, in all the neurons now in frontal cortex, because that's what we have limited here. So for example, if we wanted to limit it to just the subclusters associated with the 2 interneuron clusters, again, I'm going to select them here. Now I've gone to the subclusters, and now I can select particular target subclusters. So for example, if I want to compare within interneuron cluster 1,1 versus 1,2, I can do that in exactly the same way and go to the scatter plot. And now we can find the markers that distinguish these two clusters. And as you can see from the naming, for example, this subcluster 1.2 is positive for complexin and reelin, and you can see reelin is enriched versus just the complexin 3 here. So if you wanted to figure out where that complexin 3 marker is, we can force it to be, again, now mapped onto the scatter plot. You can see it's highly expressed in both subclusters here. So I hope that's helpful to give you a sense of how you can use this set of tools by choosing target subclusters versus particular comparisons to find markers in any arbitrary combination. And in another tutorial, I'll show you how you can extend this to make synthetic clusters by summing different clusters up or comparing across tissues. So, thank you.